Hello guys and welcome back to Medu. Today we are going to be learning the topic of mechanism of cell injury. If you haven't seen our previous video on what are the causes of cell injury, please make sure you see that because it lays the foundation to understand this video. So before we begin, I'd like to ask you guys to leave a like and subscribe to the channel as it really helps us a lot and motivates us to make these videos. All the notes will be provided for free of cost in our app down below, so make sure you download the app today. So now, firstly, we're going to see how decreased ATP production results into cell injury. As we all know, ATP is required by all our cells. So if there's a cell injury caused due to hypoxia or by chemicals, it may lead to decreased ATP production. Decreased ATP production is due to the decreased oxidative phosphorylation occurring inside the mitochondria. So what are the effects when this happens? Number one, the sodium pump present in the cell membrane will fail. That's our first point. Now, second is that there will be increased anaerobic glycolysis. Now, the third point will be the failure of calcium pump. And fourth and finally, there will be failure of protein synthesis in ribosomes. Now let's understand in depth how it happens. So for instance, if this is a blood vessel, so if there's some injury to it or some part of it has broken, it will result into ischemia. Now when ischemia occurs, it will directly result into the decreased oxidative phosphorylation taking place inside the mitochondria. Now, since there is a decreased oxidative phosphorylation, it will eventually lead to decreased ATP production inside the cells. Now, due to this decreased ATP production, there are three main things that are happening. Number one, the failure of sodium pump. Number two, increased anaerobic glycolysis. And number three, the detachment of ribosomes. Now, when the Na plus pump fails, it results into the increased influx of calcium ions, water, and sodium ions. And this will also result into the efflux of potassium ions. This will eventually result into the swelling of endoplasmic reticulum and loss of microvilli blebs. Now, due to the increased anaerobic glycolysis, the three things will happen. Number one, there will be decreased glycogen present inside the body. Number two, there will be increased lactic acid levels. And number three will be decreased pH, which will eventually result into the clumping of nuclear chromatin. Detachments of ribosomes will result into decreased protein synthesis. Now we're going to be seeing the second point, which is mitochondrial damage. Mitochondria are actually sensitive to almost all kinds of injurious stimuli. So when there is mitochondrial damage, it results into number one, decreased ATP, number two, formation of free radicals, number three, formation of mitochondrial permeability transition pore, and finally the leakage of mitochondrial protein. So now what happens is when these mitochondria are exposed to toxins or radiation, it results into mitochondrial damage or dysfunction. So when these get damaged, two things will happen. Number one will be decreased ATP generation, and number two, increased free radical generation. Now, these both together will result into multiple cellular abnormalities. And now this finally will result into the necrosis of the tissue. Next, when there is DNA or protein damage, there will be two things that will happen. Number one, it will be the increase in pro-apoptotic proteins. And decrease in anti-apoptotic proteins. Now you may ask, what are these things? Now, pro-apoptotic proteins are the proteins that increase apoptosis, whereas the anti-apoptotic proteins are the proteins that decrease the apoptosis. So next, there is leakage of mitochondrial proteins. So this will result into apoptosis. So now you may wonder, why is there a leakage of mitochondrial proteins? Well, if you remember, I had told you that there is a formation of mitochondrial permeability transition pore. So this leakage is due to those formation of those pores. So now we move on to the third point, which is the influx of calcium and the loss of calcium, homeostasis. Normally, the concentration of the cytosolic calcium is low and most of it is hidden away in the mitochondria in the endoplasmic reticulum. But when ischemia and certain toxins are present in the body, it results into the increased in cytosolic calcium levels. Initially, it is due to the release from the intracellular stores and later due to the influx across the cell membrane. Now, these levels that increase are initially due to the release of the intracellular stores and then later by influx across the cell membrane. So let's understand this in depth. So let's take this calcium ion into perspective. This is a calcium ion present in the extracellular fluid. So when there is an injurious agent acting, it will result into the influx of this calcium ion from the extracellular fluid to the intracellular fluid. Once this calcium ion reaches inside, it, along with the mitochondria and the smooth endoplasmic reticulum, increased level results into the increased levels of cytosolic calcium levels. Now, this increased cytosolic levels will result into the activation of cellular enzymes and the increased mitochondrial permeability transition. Now, these cellular enzymes include phospholipase, protease, ATPase, etc. There are many more enzymes. I have not mentioned them right here, but there are many more enzymes that are involved. So, this activation of cellular enzymes results into the membrane damage and nuclear damage. The increased mitochondrial permeability transition results into decreased ATP formation. Activation of cellular enzymes are also partially responsible for this decreased ATP formation. Now we move on to the next point, which is the accumulation of free radicals. Free radicals are unstable compounds present with a single unpaired electron in their outer orbit. Normally, these free radicals are produced and they are rapidly destroyed. But when these free radicals react with any molecule, they convert that molecule into a free radical and initiate an autocatalytic reaction. Excess free radicals can be due to their increased production or ineffective degradation. Now, the pathological effects of these free radicals is number one, lipid peroxidation in membranes. Lipid peroxidation causes extensive membrane damage. Number two, cross-linking and oxidative modification of proteins. This damages the enzyme and causes abnormal folding of proteins. And number three, and finally, it causes damage to the DNA. Now, this is the final and the most important point, that is, free radicals can cause both necrosis and apoptosis. So guys, that's it for today. Thank you for watching today's video. I hope you guys enjoyed it. So don't forget to leave a like, subscribe to the channel, and all the notes will be linked in the description and will be available in our app. Thanks for watching Medu.